Thank you all for joining us today. I'm Christopher Soroshek, and it's my great pleasure to introduce our speaker today, Dr. Isidore Losos. Dr. Losos earned his medical degree at the prestigious Hadassah Medical School, which is part of Hebrew University in Jerusalem. He also completed his residency in internal medicine and then his fellowship in hematology. He then enjoyed an incredibly successful postdoctoral fellowship at Stanford University, where he helped build our understanding of diffuse large B-cell biology, diffuse large B-cell lymphoma biology and genetics. He then moved to University of Miami, where he is currently director and endowed chair of the lymphoma program within the Sylvester Comprehensive Cancer Center. His translational laboratory focuses on lymphoma biology and therapeutics, a field in which Professor Losos has published over 300 primary research articles and reviews, a few of which I was fortunate enough to contribute to while a graduate student in his laboratory. Isidore has received a multitude of awards for his research achievements, including most recently the 2020 Sylvester Comprehensive Cancer Center Outstanding Cancer Research Award. We're so pleased that you could join us today, Dr. Losos, and look forward to hearing about your latest research. Before we start, I just want to encourage the audience to visit the website listed at the bottom of the title slide to get updates on the Connect to Science seminar series, including information about upcoming speakers. Also, during the presentation, please post any questions that you may have to the Q&A section at the bottom of the screen instead of the chat box. I'll now turn things over to Isidore.
Chris, thank you very much for the invitation and nice introduction. It's a pleasure to be, unfortunately, virtually at this session, but that's how our life changed. And we do what we do. I would be better to be in person in Boston, but things changing. So during the next 45 to 50 minutes, I will take you through a journey, my journey or my lab journey over the last 20 years that is entitled HGAL, an adapter protein regulating motility, B-cell receptor signaling, and lymphomagenesis. This is my conflict of interest. There is really nothing to declare except funding for the research. So, as Chris mentioned, when I was at Stanford doing my postdoctoral fellowship and doctoral lab rotation, I participated in the study that almost everyone in the lymphoma field knows, study led by Asha Lizadeh, that discovered that diffuse large B-cell lymphoma is in reality not one entity, but at least two different entities, that we define them as germinal center, diffuse large B-cell lymphoma, and activate B-cell lymphoma in orange and in blue here. This lymphoma look under slide identical, but they have different biology and they have different clinical behavior and different prognosis, as was confirmed by many, many studies. When the study was completed, my interest was directed to specific genes that were defining one or second of this entity. And my lab in reality till now work on several of these genes. For example, LMA2, limb domain only protein that I will not be discussing. But in this presentation, I will be discussing a protein that was really not known at that point of time. It was an EST clone 814622. That when we performed this gene expression study, our attention was directed to this EST because of the following. This EST was specifically expressed in germinal center lymphocytes. Its expression was variable across diffuse large B-cell lymphoma that paralleled but was not identical to the subdivision to GCB and ABC subtypes. It was expressed in follicular lymphoma in green, but it was not expressed in any of the non-germinal center derived cells or tumor at that time. I decided to clone this gene, and this gene was termed human germinal center associated lymphoma gene that was located in chromosomes 3q13, was encoding 178 amino acid protein with 51% identity to mouse germinal center protein called M70 that was cloned by Klaus Rajewski. That was really nothing known on the function of this protein. Klaus generated a mouse model in which he knocked down this gene, but couldn't identify the function of the gene. And when I'm analyzing the structure of the gene, what we can learn were only few things. There was a motif, I, E, and V, between amino acid 170 and 110, that was potential contact site for SH2 domains. The protein contained a modified immune tyrosine activation motif that is usually expressed in proteins that are associated with B-cell or T-cell receptor signaling. And it contained a PDZ and GRB2 binding sites. And indeed, this item motif that contained the four tyrosines and additional two tyrosines that were present in the protein could be phosphorylated as we during the journey showed by doing in vitro kinase assay. And phosphorylation was mediated by proteins SYK and LIN, two proteins that are involved in B-cell receptor signaling. Another interesting part that turned our attention to this CST was that as a single RNA expression or as a protein, this gene was also associated with survival of diffuse large B-cell lymphoma patients treated either with CHOP therapy or R-CHOP therapy, as shown on this slide. And tumors that express high expression of this protein were associated with better prognosis compared to patients that express tumors with no or low expression of this protein and RNA. And it was confirmed in an independent cohort of patients. This was independent from the clinical prognostic factors. It paralleled the GCB ABC subdivision, but it was not identical to this ABC subdivision because HGAL is expressed in many of ABC tumors as well, and not only in GCB tumors. When we analyze the expression of this protein and RNA in different tissue, again, the gene was expressed mainly in germinal center, as is shown in different tissues. There was some expression in spleen, surprisingly some expression in thymus that we could not at this point of time understand, and potentially thought that maybe it's expressed in B cells in the thymus. And once again, in cell lines, it was expressed in germinal center cells and only in cell lines derived from germinal center lymphocytes. Expression in different lymphomas was variable, not expressed in lymphomas that are not derived from germinal center, and expressed in different levels in lymphomas that derive in germinal center, like follicular cell lymphoma or diffuse large B-cell lymphoma. Since at that point of time, it was already clear to me that this gene is very interesting and I would like to understand its function. We generated a monoclonal antibody using a hybridoma methodology and confirmed that at protein level, HGAL is expressed in germinal center at a slightly higher level in the dark zone. It was a protein that was expressed on the cytoplasm, but also on the membrane of the cells. And once again, during protein arrays, we confirmed its expression in normal germinal centers, in germinal center derived lymphomas. This is follicular lymphoma, showing expression of CD10, BCL2, and HGAL, but it was not expressed in non-germinal center derived tumors. For example, in marginal zone, the expression can be seen only in residual germinal centers, not in plasma cell neoplasms, and not in myeloma and other tumors. An interesting observation, the additional and interesting observation that was observed was that when you analyze the expression in Hodgkin lymphoma that are considered to be derived from germinal center cells, but usually lose expression of majority of their B-cell characteristic genes, germinal center origin, HGAL was still expressed in Hodgkin lymphoma, as if you can see on this slide, in significant proportion of the cases. And again, expression of HGAL was an independent predictor of better survival in patients treated with standard therapy, ABVD, and this is American cohort, and this was confirmed in independent Brazilian cohort of patients. Because I'm a clinician and not only scientist, we wanted to understand better, maybe we will get some clues on the function of the gene from clinical observation. And the two clinical observations that we made at that point of time were that HGAL expression is more frequent in tumors that present with limited disease, both in diffuse large B-cell lymphoma and Hodgkin lymphoma. And HGAL expression is less frequent in extranodal tumors and more frequent in nodal tumors, suggesting that maybe this gene may be associated with some degree of regulation of tumor dissemination. During the years, we analyzed what regulates the expression of this gene in germinal center and lymphocytes, and we observed that IL-4 and IL-15, two cytokines that are expressed in germinal center, increase expression of HGAL. CD40, a stimulation marker that is usually leading to termination of the germinal center reaction, decrease the expression of HGAL. BLIM1, a transcriptional factor that is very important in differentiation of germinal center cells, the plasma cells and memory cells, also repress expression of HGAL. MIR155 degrades HGAL RNA, and FBOXO10 leads to protein degradation following PCR signaling by inducing ubiquitination of the protein. Now, because of the presence of the IP motif, it is suggested to us that maybe this gene will be also involved in regulation of B-cell receptor signaling. And as I already showed you, it was indeed phosphorylated by Syk and Lin. What we know from B-cell receptor signaling biology, that usually upon stimulation, B-cell receptor that is sitting in the non-raft portion of the membrane moves to the raft portion of the membrane where Lin is usually constitutively present and the signaling initiated in the raft part. And we wanted to understand if HGAL regulates B-cell signaling, at least at biochemical level. And to address this, we performed a number of experiments that I will summarize very, very briefly. But what we demonstrate in reality, that expression of HGAL indeed augments biochemical signaling through BCR by increasing phosphorylation of Syk, increasing phosphorylation of BTK, phospholipase C gamma, and many other downstream proteins involved in the cascade, and knockdown of HGAL, in contrast, decrease the BCR signaling activity. Next, we wanted to see if it involves other parts of BCR signaling, and indeed, BCR signaling usually induces calcium mobilization, and in the presence of HGAL, calcium mobilization was increased as well. Eventually, the mechanism underlying effect of HGAL on BCR signaling was explored and identified, and we showed that HGAL colocalizes and binds to Syk, and it does it through its N-terminal domain, and by binding to Syk, it increased Syk kinase activity, as is shown in this in vitro kinase assay, and the binding is direct binding. And what we demonstrated, I will not go into detail, but to summarize the discovery, that upon BCR signaling, now it's not playing well, in reality, antigen binds to the B-cell receptor, moves to the lipid rafts, and in the lipid rafts, there is an activation of BCR signaling by phosphorylation of Lin that phosphorylates Syk, and in the presence of HGAL, HGAL is augmenting Syk phosphorylation, and then it's dissociated from the lipid raft where it's located and degraded. In addition to biochemical function of BCR signaling, there is an anatomical function, because part of the BCR signaling is internalization of the antigen and presentation of the antigen on the surface of the cells. So B-cell activation by antigen on the surface of B-cell represents a multi-step process On initial contact with antigen, PCR microclusters form, driving B cell spreading on the surface of antigen-presenting cells. During the spreading, microcluster with active BCR signaling are moved toward the central cluster, forming what we will call central SMAC or immunological synapse, which acts as a platform for antigen internalization. And all this process is regulated by cytoskeleton. And this is a cartoon summarizing what I showed you in the words. So once again, there is formation of microclusters, then there is spreading of the cells, there is B cell contraction and formation of the signalosome portion or central SMAC, and then antigen internalization. And as you can see, in reality, all this process is regulated by SIC and LIN that are part of the BCR signaling and that with which HGAL is interacting, but also by cytoskeleton. And our question was whether HGAL really regulates the cytoskeleton portion and is involved in this signalosome formation. To address this, we generated functional supported bilayer functionals with anti-IgM, in which we took 2 layers of lipids and generated lipid vesicles into which you could insert antigens of your interest, ICAM, or for example, anti-IgM. And this was a functional membrane because this protein could move upon interaction with the cells through, and you could see the location of proteins of interest during this process. And that's what is happening. And I hope that you can see the movie. Upon stimulation, BCR is growing and forms the central SMAC area, and there is a peripheral SMAC area. HGAL is located in this central SMAC area and co-localized with B cell receptor. When you mutate the tyrosines that are present in the item motif, in reality, HGAL is currently not localizing into the central SMAC area or signalosome, and also the B cell receptor localization and formation of this signalosome part is disturbed. The area is not so condensed and it's not so functional, as is shown here on this slide. And this is confirmed in multiple cell lines, and it's also been demonstrated that HGAL regulates not only the anatomical formation of this central SMAC, but it also regulates the dynamic of central SMAC locations. And during this process, and I will not go into detail, there is an important interaction of HGAL with GRB2 that is regulating this process. So in summary of what I currently showed you, we demonstrated that HGAL regulates B cell receptor signaling. It potentiates signaling cascade biochemically, and it modulates the formation of immunological synapse. And we are currently studying also antigen internalization and antigen presentation. In addition, I mentioned that there was a clinical association that HGAL expression is associated with more localized disease and lacks frequent extranodal involvement. And this suggested that HGAL potentially may regulate cytoskeleton, and by doing that, it can regulate germinal center lymphocyte motility. So what do we know on germinal center lymphocyte motility? In reality, not a lot. Recent studies reported restricted motility of germinal center lymphocytes in the germinal center compartment that may be necessary for successful completion of the germinal center reaction. The mechanisms regulating germinal center lymphocyte motility are largely unknown. This contour shows a germinal center with dark zone and light zone, and what we learned that there is motility of the lymphocytes during differentiation process from the dark zone to the light zone, where they interact with antigen presenting cells. Antigen selection is undergoing, and then some of the cells will go back to the dark zone for additional round of mutations. But in reality, usually during the germinal center reaction, the lymphocytes almost don't leave the germinal center microenvironment. But we also know that the lymphocyte motility is a multi-step process that results from coordinated reorganization of actin myosin cytoskeleton that is regulated temporally and spatially by several proteins, but especially by Rho family of small GTPases. And Rho family activation may contribute to migration inhibition. So you may be surprised by inhibition because in many solid tumors, in reality, Rho family is contributing to dissemination. It depends on the extent of the activation of the Rho, as I will show you. An initial clue of potential role of HGAL in regulation of lymphoma cell motility came from a simple study in which we stimulated the lymphocytes with IL-6. And during this stimulation, we observed changes in the structure of the cells, formation of the podosome. And in this podosome, HGAL co-localized with actin, but that suggested that indeed this protein may be interacting with cytoskeleton. And indeed, when you modulate expression of HGAL in many different types of cell lymphocytes on non-lymphoid cell like HeLa cells, you can observe the following, that upon expression of HGAL, cell motility is decreased. And when you knock down HGAL, cell motility is increased. And there are specific areas of the protein of HGAL that I will not go into detail that may contribute to this process. In vitro studies demonstrated that HGAL expression regulates both cell movement distance. HGAL is an inhibitory protein on cell motility, and it also regulates speed of cell motility. But these were in vitro studies. How we translate this finding to in vivo studies, and how we can recapitulate what we observe in humans with decreased dissemination of the tumors. There are in reality not a lot of tumor models, of lymphoma tumor models, which can be used to assess cell dissemination or lymphoma dissemination. And we don't like this term metastasis, but that's in reality spread of the tumor. So initially, to assess the potential role of HGAL, we went to the solid oncology area and we took a breast cancer cell line 4172 that is known to specifically metastasize to the lung. And these cells definitely don't express HGAL, but we modify the cells and induce expression of HGAL. The cells were injected intravenously, and we followed formation of lung metastasis. As you can observe on this slide, there was engraftment of the tumors at 1 week post-injection, and then there was different rate of tumor growth. With animal that express HGAL has a decreased number of metastases, as you can see on this picture and on this graph, compared to the animals that did not express HGAL, indicated that potentially there is an effect on dissemination of this type of tumor. This phenomena was also could be observed in vitro because the invasion in 3D dimensional invasion assay was decreased by HGAL. This effect was not mediated by cell proliferation, as we showed, and it was not mediated by other processes, as I will show you in a moment. So that suggested to us that maybe the same thing is happening also in lymphoma tumors. And to address this, we generated several models of HGAL in lymphoma in which isogenic cells were engineered either to express HGAL or not to express HGAL. And in all the models, we observed the same phenomenon. Once you inject the cells with HGAL or without HGAL, engraftment occurs at 1 week. And after that, cells that express HGAL or tumor that express HGAL, the tumor volume in the animal is increasing at higher rate than in animals that don't have HGAL. And this is usually mediated, not usually, always mediated, by difference in the number of tumor deposits that you can find in the animals in different locations, as shown in C portion of this graph. Usually animals with HGAL usually have 1 or 2 tumor locations, but animals without HGAL have multiple localization of the tumor, and this translates to a different survival, with animals with HGAL expression without any therapy having significantly better survival compared to animal that do not express HGAL. In reality, reproducing the clinical observation that we observed in our initial clinical studies. And this can be also observed, the same phenomena when you knock down HGAL, but the same effect. HGAL is associated with lower tumor volume and better health. As I already mentioned, long-term studies to analyze if this effect is mediated by difference in cell proliferation demonstrated that there was no difference in cell proliferation between tumors with and without HGAL. Some of these curves, they are overlapping, but this is curve corresponding to the same type of cell. This observation was very, very important for us because in reality reproduced what we're observing in humans, but we wanted to see if indeed cell motility in vivo affected by HGAL. And once again, studies of cell motility are very, very cumbersome and usually done with transgenic animals. But we did not want to go this way, and we designed a different methodology that can be used to assess cell motility. To this end, what we were doing, we were injecting the cells with and without HGAL expression into anterior chamber of the eye. And the animals were kept alive, and we contract cell motility in the eye in this animal, and we contract also egress or exit of the cell from the eye to other tissues, to lymph node, to bone marrow, spleen, and so on. And this is an example of what you can see. This is injection 1 day, and you can see a lot of cells that express the dye with which the cells were labeled. After 2 days, there are already less cells. And during this day, you can track the cell motility. And after 4 days, even less cells, and you start to detect the cells outside. Now, when you inject simultaneously into the same eye, both HGAL positive cells and HGAL negative cells that are labeled by different colors, you can compare in the same eye, in the same experiment, the motility of cells with and without injection, expression of HGAL. And you can see definitely that after 3 days of injection, there are still a lot of cells that expressing HGAL in the eye, that there is only minority of the cells that do not express HGAL that stay in the eye. And this is reflected by the egress index and density that you observe over time. Marked decrease in density of the cells in the eye chamber that is significantly decreased more in HGAL negative cells compared to HGAL positive cells, and the egress of the cells or exit of the cells from the eye is again significantly higher or more pronounced in HGAL negative, confirming that indeed HGAL regulate the effects of tumor dissemination by regulation of cell motility. Next, we wanted to see what are the mechanisms by which HGAL does this. So if you take cells in vitro migration assay and express HGAL, definitely spontaneous migration of the cells is decreased. IL-6 is decreasing cell motility, and in the presence of the HGAL, there is even further decrease in cell motility, and SDF, in contrast, increase lymphocytes in lymphoma cell motility, but HGAL still decreases. So HGAL affects multiple stimulation of multiple chemokines effect on cell motility. And this effect is also can be seen not only in motility, but also in invasion of the tumor in 3D sphere invasion assay. And again, in the presence of HGAL, invasion of the HGAL in the surrounding microenvironment is decreased. But the effect of the HGAL on the invasion is not due to regulation of metalloproteases that are involved in this process. It is due still mainly due to effect on cell motility. An elegant experiment that I will not show, but were published, what we demonstrated, that HGAL regulates motility by multiple mechanisms. In our first experiments, we showed that HGAL directly interacts with both actin and myosin. It increases binding of myosin to F-actin. It inhibits the ability of myosin to translocate actin by reducing the maximal velocity of myosin head actin movement. And there was no effect on actomyosin ATPase activity. Furthermore, show you junk in my lab demonstrated that HGAL regulates ROA activation pathway by binding to two proteins, PDZ-ROGF and large PDZ-ROGF, activating ROGF, and by activating ROGF activity, there was an effect on downstream effector, ROC, cofilin, and other proteins, and eventually reorganizing cytoskeleton and decreasing cell motility. One of the most important processes in B-cell biology and lymphomagenesis is, in reality, as I mentioned, is B-cell receptor activity. But we almost don't know anything on the effect of B-cell lymphoma, B-cell receptor stimulation on lymphoma on normal cell motility. In previous studies, it was demonstrated that if you activate B-cell receptor in normal lymphocyte, cell motility is increased. The same effect was observed in chronic lymphocytic leukemia, but there was no studies conclusively demonstrated what B-cell receptor activation causes in diffuse large B-cell lymphoma. Studies that addressed the role of B-cell receptor in lymphocyte motility suggested that by activation of BCR, there is an activation of phosphatidylenoacetyl kinase that activates ROA, and that's really regulates cell motility. To our surprise, in contrast to what was present in the literature with CLL and normal lymphocytes, we observed that upon B-cell receptor stimulation, there is a decrease in lymphoma cell motility in vitro. And this decrease in cell motility in vitro is enhanced by HGAL that enhance BCR signaling, suggesting that BCR signaling in diffuse large B-cell lymphoma is decreasing lymphoma motility and maybe in reality decreasing lymphoma dissemination. Similarly, if you will stimulate the cell with SDF, one of the most important chemokines, it will increase lymphoma, diffuse large B-cell lymphoma cell motility. HGAL decreased this effect of SDF, but combination of HGAL and IgM decrease it even slightly further, depends on the cell. What is also important, as I already mentioned, in many cells, activation of ROA suggested by these previous studies would lead to increased cell motility. But that's not the case in lymphoma. In lymphoma, by using constitutively active ROA that we express in these cells, there was significant decrease in cell motility, suggesting that there is a very tight regulation of ROA activity in the cells, and both very high activity or very low activity of these proteins may decrease cell motility. And this is shown in reality by in this slide that in the presence of IgM stimulation, you can increase ROA activation, but this translates to decreased cell motility. And when you knock down HGAL, there is a decrease in ROA activation by IgM, and this translates in increased cell motility. We next wanted to understand what's the interaction of different mechanisms and where HGAL stands in the array of signaling pathways that regulate cell motility. To address it, we used either knockout or knock-in experiments or using specific inhibitors in which we inhibiting actin polymerization by using latricolin, knocked down ROA, stimulated the cells with SDF1, inhibited SYK by using R408 inhibitor or inhibiting BTK. In all this experiment, we observed the following: that in reality, HGAL is located up in the signaling pathway above actin, above SYK, above BTK, and above ROA, confirming our other experiment that activity of HGAL is in reality very close to the cell membrane, above these internal mechanisms, intracellular mechanisms that regulate cell motility. So it's a very early activator or regulator of cell motility in the cell. Now, we showed multiple mechanisms, but do we really know what if this is the all mechanisms that exist or this only partial? So we wanted to understand better the proteomics of HGAL interaction in the cells. And to this end, we used in bioID proximity-dependent protein biotinylation methods, in which you can express protein of your interest, in our case HGAL, to which you link E. coli enzyme that is called BR asterexis, that can induce biotinylation of protein bounds of this protein or proteins in the close proximity. And once the proteins are biotinylated, you can pull down the proteins and perform mass spectroscopy and identify who are the proteins in close vicinity to HGAL or in direct interaction. And when we performed it, we in reality were surprised that HGAL interacted with 96 proteins as potentially interacting proteins, but majority of these proteins, 30 unique proteins, were part of the cytoskeleton. And you can see the goal enrichment in the different functional properties of the cells, for example, cytoskeleton, actin binding, lamelloprotein. Multiple interactions are involved in regulation of cell motility, and a single adaptor protein can regulate many of these processes. An interesting observation from this study was that HGAL interacts with tubulin, both with alpha tubulin and beta tubulin. And we confirmed by using an IP experiment that interaction indeed happens, and by using recombinant tubulins and recombinant HGAL proteins, we confirmed that this interaction is direct. The interaction was occurring in the peripheral SMAC area, not in the central SMAC area, and peripheral SMAC area, where majority of the cytoskeleton proteins during B-cell activation are located. And once again, if you now inhibit tubulin function, either by vinblastine or by colchicine, you can demonstrate that this is tubulin function is downstream in HGAL because it eliminates HGAL effect on cell motility. So in summary, HGAL inhibits human B-cell motility by multiple mechanisms. It only shows us that when we try to simplify our understanding of motility by analyzing a single effect of the gene or protein, this is not the end of this activity of this specific protein. And we demonstrated that HGAL regulates actin myosin cytoskeleton by directly interacting with actin myosin. It regulates ROA signaling. It interacts with tubulin, and it interacts with our components that also regulate cell motility. And in additional, approximately 25 proteins that we still did not touch, but still most probably will be involved in regulation of cell motility. However, the next question that we wanted to ask was, because HGAL regulates both B-cell signaling and because HGAL regulates now cell motility, can it contribute to lymphomagenesis? During the recent years, we learned that several proteins that are mutated, or genes that are mutated in germinal centers, are in reality proteins that are regulating cell motility. The best example of this is GNase13, SR1PR2 proteins, which were shown to be mutated, especially in germinal center tumors, diffuse large B-cell lymphoma, and these mutations led to lymphomagenesis when animal studies were performed. To address this, we generated 3 conditional mice strains in which we expressed HGAL under control of different promoters. Stem cell promoter in hematopoietic stem cells, SCAL1, MB1 promoter at the level of pre-B cells, or in germinal center under the control of AID protein. The expression of HGAL in this animal was as we would expect. HGAL was expressed only on induction of germinal center reaction in AID animals, while it was expressed in all the hematopoietic cells in SCAL1 animals. When we analyze these animals, we observed that in animals in which HGAL is specifically expressed in germinal center, there is an increase in number of germinal center lymphocytes. This is by flow cytometry data, but this is a summary, suggesting that HGAL may contribute to the size of germinal center reaction by simply, most probably, inhibiting egress of the cells from germinal center. And indeed, when we analyze this also by immunohistochemistry, we observed the largest germinal centers in HGAL expressing animal under the controls of AID. To our surprise, when we followed this animal for long term, we observed that starting from approximately 12 months, all the three models started to develop tumors. And some of these tumors were lymphomas that were occurring in approximately 20% to 30% of the animals. When we analyzed these lymphomas, they were diffuse large B-cell lymphoma tumors, and their immunophenotype was a germinal center phenotype corresponding to specific expression of HGAL in germinal centers. The tumors were clonal by analyzing immunoglobulin rearrangement. And more importantly, when we analyzed also mutational pattern of these tumors in animals, they expressed many proteins of many genes that were mutated, and this is the same genes that we observed mutated in germinal center derived human diffuse large B-cell lymphoma. By analyzing the gene expression of this tumor, there was a similarity of expression of this tumor to what we observe in follicular lymphoma, that is another tumor of germinal center origin. And there was enrichment of genes originated in expression of germinal center and MYC target genes in induced tumors, once again suggesting that these tumors that are generating in HGAL overexpressing animals are indeed germinal center DLBCLs. So in conclusion, HGAL is a specific protein that is specifically expressed in germinal center. It inhibits human B-cell lymphoma motility, both in vitro and in vivo, and most probably, based on our data in vitro and vivo and clinical observations, regulates tumor dissemination in the human beings, in the patients. HGAL regulates raw A and B cell signaling processes, as we demonstrated. It plays a role in germinal center reaction, and it contributes to larger germinal center and more exaggerated germinal center reaction. And in our recent studies, we demonstrated that HGAL, similar to GNA and ESA1, PR2 proteins, other proteins regulating germinal center motility, may serve as a B-cell oncogene. So I want to cite at the end of my talk, Oscar Wilde, that once said, the moment you think you understand a great work of art, it is dead for you. I'm working on this protein for 20 years since I have cloned it. And the protein still surprises us and still leads to unexpected findings. And I cannot finish the work on this protein. And I will finish my lecture with the following finding that we recently published in Pathology Journal. Well, through all these 20 years, we thought that HGAL is a germinal center specific tumor, but we still kept in mind that we observed some expression of RNA in the thymus. More recently, we demonstrated that HGAL is also expressed both in normal and neoplastic follicular helper T cells, including angioimmunoblastic T-cell lymphoma, suggesting that similar to many other germinal centers, genes that are expressed in these specific types of cells and tumors, HGAL is also expressed in these tumors and normal cells, and most probably has some function in these T cells that we till now did not touch. This work was done by many people during the course of the last 20 years. Some of the people are still with me in the lab, Xiaoyu Jiang and Chengxin Lu, that did a lot of work on this protein. Vincent Moy is my collaborator from biophysics that enabled us to do the very nice microscopy studies and establish the lipid raft vesicle model. Mithat Abdelreda is the person from Miami Institute of Diabetes that help us with in vivo motility studies. And all other people were people in the University of Miami that at a certain point of time touch HGAL at different stages. I collaborate with Isidro Sanchez Garcia and his lab, with which we work together to generate the transgenic animals, and with Jose Martinez Clement from Pamplona. And since I already mentioned that I cloned this gene when I was postdoc in Ron Levy's lab, many people who worked with me at Stanford at that point of time continue to work and collaborate with us on this project, including Yasonet Panam, pathologist Andrew Gentles, Asha Lizarel, and statistician Rob Kipsharon. Thank you very much.
Thank you, Isidore, for that wonderful talk. You've done some not only incredibly important biology and gained some new insights into lymphomas, but also have innovated in some really interesting ways during that process. And I was really taken aback by some of your approaches, doing some very interesting and innovative migration assays and colonization. So I thought that was really neat. So if audience members can type their questions into the Q&A section at the bottom of the screen. And while they're doing that, I do have a couple of questions. First, you show very convincingly that HGAL has multiple roles in B cell biology, B cell migration, as well as B cell receptor signaling. So can you, and then you showed at the very end that when you overexpress HGAL in mouse models, they do develop lymphomas. And do you have a strong sense of which part of HGAL biology or its effects is mediating that increased lymphomagenesis? Is it the B cell receptor signaling? Is it contributing to migration of cells somehow where they can move to areas of increased survival or proliferation? Do you have a speculation about what it is that the mechanism that's driving the lymphomas?
As you say, speculation and some data, but I think that, as a scientist and as human beings, we are always trying to simplify things. And we want to present a clear picture of one mechanism and one schema in the manuscript, and editors and reviewers very, very frequently like to have things are very simple as much as possible. Unfortunately, biology is more complex. And we think that both processes that you mentioned, both B-cell signaling and motility, are interlinked and both are contributing to lymphoma development in this case. As I already mentioned, there are several other genes that when they're mutated and affect cell motility, they also lead to lymphomagenesis of germinal central-like tumors, suggesting that continuous presence of the germinals of the B cells in the germinal central microenvironment may be detrimental for pathogenesis, because these cells are sitting in the microenvironment with high mutational activity and undergoing multiple rounds of mutations that eventually lead to mistakes and to lymphomagenesis. But at the same time, B-cell receptor signaling is very, very important in many ways, because we know when you knock down B-cell receptor, you don't have lymphomas that work plus Rayevsky. So it's a combination of enhanced activity of BCR, continuous presence in the same microenvironment without ability to leave the same microenvironment, and potential exposure to mutational activity of germinal center by AID-mediated mutations, all of which contribute to this lymphomagenesis.
So I suppose those multiple factors and multiple pathways that HGAL is contributing to, or pleiotropic effects, will make it somewhat of a challenge to pursue this therapeutically. But what can you say about the potential to drug HGAL in a way for as a strategy for lymphoma therapy?
I don't think that HGAL we will be able to drug because once again, still HGAL is associated with better prognosis. But we are using HGAL as a model to understand mechanisms underlying these processes, and these processes can be druggable. So it's the underlying mechanisms that are downstream of HGAL. These are the ones that can be druggable and can eventually result in improved outcome. There was a science paper that went maybe unnoticed by many people in many fields, but it was a very, very important paper, which once again, they performed a knockout assays for multiple genes and analyzed what effect they have on lymphomagenesis and on lymphoma progression. And one of the most important genes that was demonstrated in this paper was actin. When you knock down actin, there was no lymphomagenesis and there was no lymphoma progression of established tumors. And that only suggests that the cells need cell motility in order to survive and in order to produce aggressive tumors. And this motility part of lymphomagenesis that is almost unknown and only few labs, including mine, are studying it, may be very, very important eventually for potential targeting and assessing this targeting effect in human beings.
Yeah, I would agree. I would think that different regions of the dark zone, light zone, all the B cell signaling that occurs there could be quite different, and the migration into areas where more pro-survival factors are being provided would be an important part of the pathogenesis. But we can move on to an audience member's question. So this is from Zintis Indy. Zintis writes, it's interesting that HGAL expression correlates strongly, but not completely with lymphoma subtypes. The mechanistic insights you presented suggest a reason that altered HGAL expression would be enriched, but not ubiquitous in certain subtypes.
It's an excellent question. The GCB and ABC subtype are defined by array of gene expression, and there is really not a single marker that can be used to precisely identify who is GCB and who is ABC lymphoma. Even, for example, BCL6 is so-called one of the master regulators of germinal center reactions that is highly expressed in germinal centers. It's also expressed in ABC tumors. So once again, it's a continuum of expression, and that's why when we divide the cells into GCB and ABC, it depends where you put the division cut, who is who. But there is a continuum of expression, and some genes are expressed more and less. What we observed, and I didn't show you here, that in reality, downregulation of germinal center genes is a continuous process. And the best example for this is a Castleman disease. Castleman disease is a disease of germinal centers in which there is involution of germinal centers. And during this process, they are losing germinal center expressions. And we showed, and I didn't mention it, that HGAL is one of the last germinal centers protein that is still expressed in Castleman disease. So it's really one of the last markers for germinal center derivation. And that's why we also see it in Hodgkin lymphoma, where majority of the germinal center markers are also. So I'm not surprised for that reason, that there are ABC tumors that a majority of the germinal center signature is already downregulated, but HGAL is still expressed.
Wonderful. Thank you for that. I think if other audience members have questions, they can reach out to you directly as well. We're going to finish up today's session, but please feel free to reach out to Isidore at his University of Miami email address. Thank you again, Isidore, for the wonderful seminar and for sharing your afternoon with us today. We look forward to continuing to hear about your updates on lymphoma genesis, biology, and signaling. Thanks again.
Thank you very much for the invitation. Thank you.
Thank you.